In part 3, we'll take a look at cellular division. So if you think about it, when we were conceived and we were one cell, that cell had to divide into two cells. And then the two cells became four cells, and so on until, as an adult, we have about 75 trillion cells. So all of our cells are actually derived from other cells, right? So cellular division is really important because we need it for growth, right, to become to the stage we're at now as an adult, and then also during times where we have to repair our body and we need to make new cells. So even though we're not growing, we do need that ability to have cellular division. The cell that's going to divide is known as the mother cell. Right? So the mother cell that's going to split in two to form two identical daughter cells. It's not as if the mother cell gave birth to the two daughter cells and the mother cell is still there with the two daughter cells. It's not like that. The one mother cell is going to divide into the two daughter cells, right? It's going to split. Now, we have two types of cellular division. The one that we're going to talk about in this lab is known as somatic cellular division, right? You may put a little star next to that or a highlight. This means we're talking about the cellular division of our body cells. Somatic is really an important word. We use this all semester long. Anytime we're talking about our body cells, any cell that's not a sex cell, right? The sperm or the egg, right? An oocyte. So somatic cell division is going to have two phases to this. The first phase we're going to take a look at is called mitosis. Right? And this is where the nucleus actually divides so that each daughter cell gets a full complement of chromosomes. Right? We usually say 23 pair, right? Or 46 chromosomes. So mitosis is when the nucleus divides, but then after the nucleus divides, the cytoplasm has to divide. So we're going to call that cytokinesis. In anatomy and physiology, too, we'll consider reproductive cellular division, as this occurs in the sex cells, and that's when we cover sex cells in reproduction. So same thing, the nucleus needs to divide, but instead of calling that mitosis, we're going to call it meiosis. Right? So it's just nuclear division known as meiosis. And here, each cell is going to have only half the genetic complement, right? They don't want to have the full 23 pair. It's only going to have half. The cytoplasm of the sperm and the egg cell also need to divide. That is known as, excuse me, that is known as cytokinesis. Okay, so like I said, we're going to focus on somatic cellular division. So before I show you the actual stages of mitosis and then cytokinesis, I want to tell you a little bit about the life cycle of the cell. In our lab book, we have a really good picture. This demonstrates the stages of a cell's life. Let's just say here a new daughter cell is born, right? It's been created. It's now going to enter a phase known as interphase, right? Interphase is the period of time that the cell is in most of the time when it's not dividing, right? So interphase, it's important to know, this is when the cell is living its life. It's not dividing. It starts here when the cell was developed, and then the cell is going to live. And again, don't worry about the hours here, but it's going to live its life, communicating with other cells, producing proteins, just doing the normal things it's doing in our body, and it will continue to live its life. At some point, though, it's going to divide. Right? Near the end of interphase, it will then get ready to go into mitosis. Right? And mitosis is designated here by the letter M. So during this period of mitosis, the nucleus is dividing. We're going to see there's four phases to make the nucleus divide: prophase, metaphase, anaphase, and telophase. I always tell students to remember PMAT, P-M-A-T. So these are the four phases that cause the nucleus to divide. Notice in anaphase and telophase, This is when cytokinesis is going to take place. So near the end of nuclear division, the cytoplasm is going to be dividing also. So let's talk about this interphase. So it's important to us to know that this is the time of a cell's life when it is not dividing. Right? Inter means between. So the way I look at it, it's between divisions, right? When it was first born and then when it's going to divide, right? So that's what inter means. Now, even though the cell may not be actively dividing, it does have to prepare for cellular division. A mother cell that's going to divide can't just split in half, because then the two daughter cells will just have half of all the materials genetically and the organelles. Before it can divide, it needs to double everything. We need to double the organelles, we need to double the DNA. Then, once everything is doubled, it can go through division. So, during interphase, it's going to start to prepare. It's going to go through a period known as G1. The main thing I want you to know here in G1 is the duplication of organelles. So all the organelles that we discussed in the previous parts, part 1 and part 2 of this lab, are going to double. All right, so we're going to have doubling of all the organelles during G1. The cell will continue to live, and then it will go into another stage known as the S phase. In the S phase, the main thing I'd like you to know here is that the cell is going to go through DNA replication. We're going to double all of the DNA. This way, again, each daughter cell will get the full complement. So once the cell has gone through G1, doubled its organelles, and then it's gone through the S phase, doubled its DNA, right before division of the nucleus, it's going to go through another phase where it's primarily producing proteins and getting ready for cellular division. This is known as G2. Okay, so these are the phases that the cell is going to go through to prepare for cellular division. You know, it's interesting. Not all cells in our body do divide. When a cell that is not going to divide, like a muscle cell, a nerve cell, those are cells that don't divide. They stay in interphase forever. We would actually call that G0. So this is a cell that's living in interphase, but is not getting ready to divide. So let's just say our cell went through interphase G1, it went through interphase S phase and interphase G2 phase. At this point now, it is ready to go. It is going to enter mitosis, and then we'll take a look at the 4 phases of mitosis, and near the end of mitosis, it will go into cytokinesis. That's a great picture to understand and study. Here's just the notes for it. So interphase, right, again, once the cell is born, it's a very active period. The cell is growing, right, just doing its routine functions, making proteins, communicating with other cells, and preparing for cellular division. However, just kind of note, if a cell performs its function but is not preparing to divide, it's just kind of living its life, they do call that G0. In G1, the main thing, if you want to just highlight, this is the main thing I want you to know. We replicate all of the organelles, right? And I'm assuming also you knew the centriole pair, right? The centrioles become important for mitosis. The S phase, the main thing I want you to know, DNA replication. And then right before division, it's going to go through G2. And the main thing that's happening here is we're producing proteins. So all of this is interphase. So now the cell is ready to divide. Let's take a look at mitosis. If you remember, this is going to be the division of the nucleus. And then you want to remember the 4 phases, P, M, A, T, PMAT. The first phase of mitosis is prophase. So let's take a look at what's happening. In the nucleus, the chromosomes start to become visible. In part 2 of this lab, I discussed in the nucleus the DNA of a cell that's not dividing is called chromatin, and we really don't see it under the microscope. But in mitosis, in prophase specifically, the DNA turns into chromosomes and they start to become visible. We see them under the microscopes, right? So the chromosomes become visible, they shorten, and they move towards the center of the cell. In the cytoplasm, the two centriole pairs, right? So in a normal cell we just have one in a cell that's not dividing, but in a cell that's dividing we have two. They start to migrate to opposite sides of the cells. And connecting the centriole pairs, we start to see some microtubules. They actually extend from one centriole pair to another centriole pair. They're going to be important a little bit later when we see the chromosomes actually splitting, right, dividing. So let's go to a picture in our book over here. What they're showing you is a cell. Actually, let's do the diagram first. It's showing you a cell that's in interphase. Again, it's living its life, right? It's not dividing. If you look in here, you can see there's just some lines in here. This represents the chromatin. We really don't see chromosomes, and it just kind of tells you that it's not dividing. Here we have one centriole pair. Here's an actual micrograph. Right? So you can see the nucleus in here. You can just see some lines in here. This is just going to be the chromatin, and there's the centriole pair over here. But notice in prophase, we start to see the DNA become visible, right? So now it's starting to become visible in here, and we also have one centriole pair here and another centriole pair here. So notice they're starting to split apart. One's heading like kind of to the North Pole here. This one here is kind of heading down to the South Pole. And if you take a look right there, those red lines going across, those are the microtubules that are starting to develop. All right, so this would be kind of an early part of prophase. But notice when we get to the late prophase, near the end of prophase, look at the centriole pair. It's on the North Pole. Here it's on the South Pole, right? It's just about on opposite ends. We have beautiful microtubules going across, and we can see well-developed chromosomes at this point. Okay, so those are the changes that we see in prophase. I forgot to just mention, here's the DNA first starting to become visible, and at this point, you could actually see chromosomes. If we kind of blew this up, you would see chromosomes a little bit better. So that was prophase. Metaphase is the second phase of mitosis. The main thing that's happening here is the well-developed chromosomes are going to migrate towards the center of the cell. What we're going to form is something known as the metaphase plate. You may want to highlight that. This is the main thing that you're going to look for. Here, the chromosomes line up in a straight line. So if you take a look here, notice the chromosomes now have lined up, forming this metaphase plate, and you can't miss it on the microscope. Look at this, it's beautiful. It's just a nice straight line. The centriole pairs are on opposite ends at this point, and again, we can't see them too well, but you can see them a little bit. You can see the microtubules going across. So this is classic metaphase. Look for that straight line of chromosomes. The third phase of mitosis is the anaphase. In Greek, ana means to separate. So the main thing that's happening here is the chromosomes are going to separate so that half is going to move towards one cell that's going to become one daughter cell, and then the other half moves to the other cell, which is the other daughter cell. So if you want to just highlight, the main thing here is separation of the chromosomes. The spindle, which are those fibers, pull apart the chromatids and just kind of split the chromosomes so they go to the opposite side. So if we go to the book, this is anaphase. You can see again the chromosomes have split. So here's one set of chromosomes heading towards the North Pole here. Here's the other set of chromosomes heading to the South Pole over here. This is a classic appearance when you see this, and you can see it down over here. Notice half the chromosomes, right, this kind of blackish area here is being pulled this way, and then these chromosomes right to the left of my cursor are being pulled to that centromere, centrioles, excuse me, centrioles. So this is, again, a classic appearance. You see this under the microscope. It's exactly what it looks like. Right? That's anaphase. In the late part of anaphase, they're not showing it here. I have it in my notes, and we'll see it on our mitosis model. The cytoplasm starts to pinch in a little bit here, and they have it labeled over here. You can see how it's starting to pinch in a little bit. It begins in the late part of anaphase. This is known as the cleavage furrow. So highlight right here, the cleavage furrow starts to develop along the metaphase plate where the metaphase plate was and begins to pinch in. Right? A furrow would be like a little bit of a depression. And I'll show you on the mitosis board model that we're going to look at in a moment. So once the chromosomes have separated in anaphase, we're now going to finish up cellular division in telophase. Right, so telophase is the last stage of mitosis. The main thing that's happening here, guys, because the nucleus has split already, the DNA has split, the main thing that's happening in this phase is cytokinesis. Right, we're going to continue to partition the cells so that we now have two new daughter cells. Each new daughter cell is going to have its own nucleus, it's going to have its own set of organelles. And if you take a look here, as telophase continues, right, it starts to pinch off. Eventually, this will separate. We'll end up with two new daughter cells. And what phase are they going to be in? They're going to be in interphase, right? They may stay like that for a couple days, a couple weeks, a couple months, right? Forever, they may stay like that depending on the cell. So when a cell is just living its life, they're in interphase. Okay, so now that we have an idea of the phases, let's take a look at this model here. In lab, we call this the mitosis board. What we see here is the cell as it goes through nuclear division, because we're talking about mitosis. The first three boxes we're going to see, 1, 2, and 3, are all prophase. Right? In this model company, this is going to be early prophase, middle prophase, late prophase. Then this one here is metaphase. Right? We're going to go through each one of these things. Here's the metaphase plate. The next two, this is early anaphase, late anaphase. This is going to be telophase. Finally, once this splits, we have our two new daughter cells, and they are in interphase. So let's take a look at the first three prophases. Here I have early prophase, middle prophase, late prophase, just to show you some of the things that are happening. So in early prophase, you can kind of see the DNA, but it hasn't formed chromosomes yet. So here's the DNA. It's starting to become visible, right? They shorten and they thicken. And then you have one centriole pair here and another centriole pair here. Notice they're close together. By the time we get to middle prophase, here's the centriole pair here. It's starting to migrate this way. And then this centriole pair here is starting to migrate this way. And look at the chromosomes. They're starting to look like chromosomes. By the time we get to late prophase, right, the centrioles are just about on opposite sides, right? They should be pretty close to being on opposite sides. And then we have beautifully formed chromosomes. All right, so be careful or be sure on a practical. If I asked you to identify this phase here, don't just put prophase, right? You would have to indicate early prophase, middle prophase, late prophase. The next phase is metaphase, right? The main thing you're going to see here, and as well as under the microscope, is all the chromosomes lining up in a straight line. Right? Just to go back to late prophase, see how they're just kind of scattered about? By the time we get to metaphase, look at these beautiful chromosomes, straight line. And at this point, look at the centriole pairs. They're on opposite ends, and we have nice, these red fibers are microtubules. Right? They're going to become important again to, right here, they're going to become important again to separate the chromosomes. So this phase is metaphase. The next one is going to be anaphase, but this is the early part of anaphase. You can see the chromosomes have separated, so this line right here, this group of chromosomes are going to be pulled to the left, and then this one right here, these to the right, are going to be pulled to the right. So then you're starting to see the separation. But this is the one that really shows it. We can see all these over here being pulled to the left, and then again on the opposite side, all these being pulled to the right. So again, ana means to separate. Now we have the beginning of cytokinesis here. Notice this little indentation right here, right where the cytoplasm is starting to pinch off. This is known as the cleavage furrow, right? And we're starting to pinch off the cytoplasm. Once anaphase is done and the chromosomes are separated, we're now going to enter telophase. At this point, look at this. We're getting a new nucleus starting to develop here, a new nucleus develop here. They don't show it, but the organelles would all be in the cytoplasm, and the cleavage furrow is going to continue, right? We can see the cleavage furrow here, and it would just kind of continue until this separates in half. All right, so that's telophase. Once this has completely split, we now have two new daughter cells which are going to go into interphase, right? So here's one new daughter cell, here's the other new daughter cell, and these are identical to the very first slide that we went over on this model, to the mother cell, right? These are just identical. This is the mother cell split in half. Okay, so that is our mitosis, cellular division and mitosis.